Hello, and welcome to the fifth segment of 5 segments on the nervous system. This recording covers slides in part 2 of 2, slides 115 to the end. In this last recording, we'll cover topics 6 and 7. We'll start with neuroplasticity, and then we'll cover just 2 of the special senses, vision and gustation. Gustation is your sense of taste. And by that point, we will have covered all of these special phenomena along the way. The phenomena that we'll talk about in this lecture recording include after images, color blindness, and the fact that wine tasting is bogus. So let's start with neuroplasticity. So the human brain is born to do many things. The human brain is born to speak any language, to potentially develop great creativity, or perhaps terrific analytical and mathematical ability, to play any musical instrument in existence, to develop any hunting technique, or to play any sport. And so as we grow and develop, as we learn, the patterns of connections between neurons form and almost solidify, like a melted plastic will become solid as it cools. And so as we grow and develop, we speak just one or maybe a few languages. We develop certain skills and not others. So brain plasticity, being born with this brain that could potentially live in any environment, is natural selection's way of meeting an environment that might change from one generation to the next. And so the degree of plasticity, the capacity for the central nervous system to change, appears to be genetically regulated. Children exhibit far greater brain plasticity or neuroplasticity than adults. And we are currently learning that the capacity for plasticity can be manipulated using certain drugs. So in the future, it might be commonplace to take a particular drug as a therapeutic tool because that drug enhances neuroplasticity. It can give rise to a new critical window where learning or unlearning becomes particularly possible. So therapies might involve unlearning a phobia, so to speak, or learning to talk again following a stroke. So neuroplasticity refers to changes in both structure and function of the nervous system that occur throughout our lives. And what gives rise to these changes in structure and function? So new synapses, new connections between neurons can form, and that occurs when cells develop new dendrites or even new axons, and that's called sprouting, much like a tree will sprout. And then older synapses can be pruned. We can cut away the branches or the connections between cells. And then the pathways of communication from one cell to the next as part of a neural network, those pathways can grow smoother and facilitate easier communication between cells, or those pathways can sort of wither and becomes more difficult for one cell to signal the next. So let me remind you that brain functions occur because of communication across broad numbers, big numbers of neurons, and these are neural networks. And so your ability to calculate, say 16 times 23, or your ability to consider the meaning of the word justice, or your ability to analyze a work of art and interpret the artist's intentions. Those abilities, the skills, are brain functions, and they are produced by groups of neurons that are connected that work together to give rise to these abilities. So neuroplasticity involves changes in individual cells, individual neurons, as well as changes in how connected these neurons are to each other, the strength of synaptic pathways, how easily two or more cells can communicate with each other. At the level of the cell, an individual neuron can change in two different ways. Number one, an individual neuron can change the expression of a receptor for the neurotransmitter. So I hope you remember that neurotransmitter, or all receptors, are proteins, and neurotransmitter receptors are receptors for neurotransmitters, and neurotransmitters are signaling molecules that occur at chemical synapses. And if the cell upregulates a particular receptor protein, then the cell becomes more sensitive to that neurotransmitter. And if the cell downregulates the receptor, then the cell becomes less sensitive to that neurotransmitter. So it's like adjusting the volume on a hearing aid, so to speak. We can increase the volume and make that cell more sensitive to signals, or we can decrease the volume and make that cell less sensitive to signals. And the second way that an individual cell can change is in the number of dendrites. And we call that either sprouting or pruning. So when a cell increases the number of dendrites by growing, by literally forming new dendrites, we call that dendritic sprouting. And then dendrites that either are not used frequently or should not be used for communication, cell-to-cell communication, those older dendrites can be pruned, where they will degenerate and the cell will have fewer dendrites following dendritic pruning. And then we can change the strength of the synaptic pathways. And one way that we can change the strength of the synaptic pathway is by modifying the abundance of receptor proteins at that chemical synapse. A second way in which we can change the strength of a synaptic pathway, or the ease with which two cells communicate, is by changing the number of synapses, the number of physical connections between cells in two different parts of the brain. If you look in the lower left corner of this slide, you'll see a link to a YouTube video. It's definitely worth 2 minutes of your time. And that is also posted in Canvas under the recommended tutorial videos. I hope you'll watch it. There is also a much longer video on neuroplasticity. It's about an hour long, and it's a wonderful video all about neuroplasticity that comes from the World Science Fair a few years ago. I hope you'll watch that one too. So what's the purpose of neuroplasticity? Why are our brains so changeable? So neuroplasticity is necessary in order for learning as well as for developing memories. So those are very different functions, and even within each category, learning and memory, there are many different subtypes. There are many subtypes of learning and many subtypes of memory. So learning is broadly defined as the ability to develop new skills or learn information through instruction or experience. So I hope you are all engaged in learning right now. And here are two examples. We have all learned many cognitive tasks, and we have all learned many motor skills. So when you applied that Nernst equation to calculate the membrane potential when a particular ion comes into equilibrium, that's a cognitive task. Or when you developed an ability to type on a keyboard, for example, that's a motor skill. Memory is broadly defined as the process by which information acquired through learning is stored and retrieved. And memories appear to be stored not in individual neurons, but rather they are stored as patterns of excitability. And so every time you recall a particular memory, so let's think about your first grade teacher. Every time you remember your first grade teacher, a particular pattern of neurons become active in your brain. And there are different forms of memory. And one form of memory that I recently learned about is called prospective memory. So how is it that you can remember that on your way home from work or your way home from school that you need to stop and get milk and you need eggs and you need bread, or you have to remember to go to the pharmacy and pick up your prescription? So prospective memory, just as an example, is one form of many different subtypes of memory. Brain plasticity is an important part of normal growth and development. And every newborn has an incredibly plastic brain in the sense that there are myriad patterns that could come into being depending upon what environment that baby is exposed to. And so when we're born, we have approximately each neuron in our cerebrum, in our cerebral cortex. That's what cortical neuron means. Each neuron has approximately 2,500 connections with other cells. And depending on what the baby is exposed to, a particular pattern of connection will develop, and any given 3-year-old has approximately, each cortical neuron has about 15,000 connections with other cells. And then notice that there's a drop in the number of cortical connections as we approach adulthood, or as we go through childhood. Significant synaptic pruning occurs. So the synapses that are less utilized are pruned back. As we reach adulthood, each cortical neuron has about 8,000 synaptic connections with other cells. And so as we go through childhood, it's as if your nervous system is figuring out, okay, what connections do I really need to be functional in this life, and what connections are sort of a waste of energy in terms of maintaining them? What can I prune back, and what do I need to reinforce? Brain plasticity is also involved in a recovery following an injury to the central nervous system. So a brain injury might come from trauma, or it might arise from infection or a stroke or a cancer. And no matter the cause, injury to the brain or the spinal cord means that there are neurons and neuroglia that have been damaged, and they are no longer able to perform their normal function of communication. And so as part of healing or recovering from that disease or that injury, brain plasticity occurs. Neural pathways can be rerouted. So parts of the brain that used to communicate with each other, but that pattern of communication was disrupted due to injury, they can find a new route of communication. Just like if there's construction, as you're traveling from one part of the city to the other and you need to take a different route in order to connect those two parts of the city, something similar happens in your brain. Only it takes some time and some repeated stimulation in order to reinforce or newly enforce certain synaptic pathways, or even just flat out generate brand new synaptic pathways. So in this diagram up top, we see the pre-injury condition, where we have a purple neuron communicating with both a blue neuron and a yellow neuron. And notice that both the yellow and the blue neuron have axons that form synapses with the purple neuron. And so down below, we see that some injury occurred to the blue neuron, and now the blue neuron no longer communicates with the purple neuron. In the absence of that communication between the blue neuron and the purple neuron, the yellow neuron will compensate by strengthening the synaptic pathway. So the blue neuron and the yellow neuron are in the same part of the brain, and the purple neuron is in some different part of the brain. And so in order to ensure that those two brain regions maintain a strong connection, the yellow neuron will sprout new axon collaterals and increase the number of synapses that it has with the purple neuron as a way to compensate for the blue neuron that has been damaged. What we've been talking about over the course of the entire nervous system is a discipline, a scientific discipline called neuroscience. Neuroscience is the scientific study of the nervous system, and we are all probably experiencing an optical illusion when you look at that particular piece of art on the cover of the Journal of Neuroscience. So for most people, that piece of art does not look like a static image, but rather it appears to be moving. And of course it is a static image. And so that's an optical illusion. It's really a brain illusion, right? The information comes in through your eyes, but your brain is what interprets the image. And your brain might be interpreting this image as if it's moving when it's really not. I wish we could cover all of the special senses, but we don't have enough time, sadly. So we're just going to cover two of the special senses, vision and gustation, and maybe you'll cover the others in a future class, or maybe you've covered them in a previous class. Things like hearing and smell and touch. We're going to cover vision and taste. So, in order to understand how vision works, we need to start with physics. We need to start by talking about light. And so we see the electromagnetic spectrum here. These are different intensities of energy, is one way to think about it. Starting on the left, we have the most intense energy rays on the electromagnetic spectrum, the gamma rays. And gamma rays are super dangerous to living things. And perhaps you're familiar with X-rays, and you know that X-rays are also dangerous to living things. This is why you have to put on the lead shield when a dentist, for example, takes X-rays of your mouth. And then ultraviolet light is also dangerous, but less so. You know that ultraviolet light can give you a sunburn and can cause skin cancer. And then look at that little cutout, that little dotted rectangle that's blown up at the bottom. Those are the wavelengths of light that we can see. So our photoreceptors, as humans, our photoreceptors can only respond to a very narrow portion of the electromagnetic spectrum. And so maybe you'll recognize the rainbow there. The smallest wavelengths are in the violet range at 400, and the longest wavelengths that our eyes can detect are in the red at 750 nanometers. And so going back to that electromagnetic spectrum, we'll continue on our journey from left to right. When we get into the infrared, that's commonly thought of as heat, and you can feel infrared radiation with your hands, but you cannot see it with your eyes. And it is generally not dangerous. Of course, heat can kill you. But it's less dangerous than ultraviolet radiation because it contains less energy. And as we move from the infrared into microwaves, microwaves are generally not dangerous to living things. Of course, we all have these devices in our kitchens that are tuned to cause the water molecules in our food to vibrate faster. And so it's a very quick way to heat up food. And then radio waves. Radio waves have really long wavelengths. They're not dangerous at all. And radio waves are around us all the time, and there doesn't seem to be any real consequence to human health and well-being from radio waves. So the major point here is that our eyes, human eyes, are able to detect and respond to a very narrow region of the electromagnetic spectrum. There are other organisms that can see in the ultraviolet range, like many birds and insects, pollinators, can see into the ultraviolet, and many flowers and fruits have ultraviolet pigments that human eyes just don't pick up. And then some organisms like pit vipers, predatory snakes, one way that they detect a prey item, like a mouse, a small mammal, is they have these infrared sensors in their face, in these pit organs. And so it's not quite the same as vision, but they can detect infrared radiation and use that as a technique for tracking their prey. So as humans, we are quite limited in our visual capabilities. And the cells that are able to respond to different wavelengths of light are called photoreceptors. Photo means light, and you're familiar with the concept of a receptor. And so we have different types of photoreceptors in our eyes, and they are located in the retina of the eye. So I'll draw your attention to the section of the eyeball shown on the right, and the retina and the optic nerve are highlighted in yellow. So the retina lines the back of your eyeball. And as we talked about in a previous lecture, we talked about how the lens will focus light onto the back of your eyeball. And those photoreceptors that are part of the retina, they will respond to and generate electrical signals in response to specific wavelengths of light. And those electrical signals, graded potentials and action potentials, are transmitted through a series of neurons, ultimately to the optic nerve. So photoreceptors transduce light energy into electric currents. So transduction. That means that one form of energy is converted into another form of energy. So you can think about a desk lamp as a transducer. It transduces, it converts electricity, right? Because it's plugged into the wall, it converts electricity into light and heat. So the photoreceptors in your eye transduce or convert light energy into electrical energy, physiological electrical energy. And the way your photoreceptors work is, of course, it's because they have proteins in their membranes, and these proteins are able to change shape when a particular wavelength of light hits them. And as these proteins change shape, it will bring about a change in membrane potential. And so these proteins, these light-sensitive proteins in our photoreceptors are called opsins. And they undergo a conformational change in the presence of light, and they are tuned to various wavelengths of light, various colors of light. On the right, we see 4 different types of photoreceptors in the human eye. 3 of them are cones, and one of them is called a rod. And on the right, we can see that these 4 types of photoreceptors have different sensitivities to different wavelengths of light. And how sensitive a particular photoreceptor cell is to a particular wavelength of light is determined by which opsins are present in the plasma membrane of the photoreceptor. So this is a common test to diagnose different forms of color blindness. So color blindness can arise from any of those photoreceptors not working properly. And most forms of color blindness are genetic in nature. So most people who have some form of color blindness were born that way, although color blindness can develop in response to disease. So for most of these circles, there is a number in one or more different colors from the background that is visible in the circle. And in class, I always caution students to take this with a grain of salt and to not be alarmed if all of a sudden they think they're colorblind because they've never seen a test like this before. And then I tell the story of my sweetie who is colorblind. He's red-green colorblind, and he didn't discover this until he was an adult, and he had already graduated college, and he was going back for a particular certification, and he needed to take a geology class. And he's super bright, he's a good student, and he is interested in science and the natural world, so of course geology class would be very fun and interesting. But he found that he struggled to classify rocks based on their appearance. And his classmates seemed to be able to do it pretty easily. And why couldn't he do it? Well, it turns out he has red-green color blindness, and he didn't know that about himself until his 20s. So, should you discover that you're colorblind, don't use this test to diagnose yourself. Rather, contact a healthcare professional. And to perhaps assuage any concerns, let's take a look at the very bottom row. So there are 3 circles in the bottom row. 2 of them just have squiggly marks in them. There are no numbers. And the one in the middle has a barely discernible number 45. And then there are actually numbers in the rest of the circles. The Exploratorium. It's so sad that our museums are closed due to the pandemic. The Exploratorium has many exhibits dealing with the perception of color. And this is one of my favorite exhibits at the Exploratorium, and it's called Disagreeing About Color. And it works best when you have a group of people. And you and your friends, or even the strangers, just the people, other people who happen to be in the museum, who happen to be looking at the same exhibit as you at the same time, a group of people can stand around and everybody makes their own decision about which of the smaller dots is the same color as the center dot. And everyone comes to a decision for themselves, and then everyone shares their decision. I think it's the dot that is at 2 o'clock. I think it's the dot that's at 9 o'clock. And people seem very, very convinced in their minds that it's definitely the one they picked. And so it just goes to show that color perception is very subjective. So all the people there are looking at exactly the same wavelengths of light. And light wavelength, color, we can measure that objectively. We can measure what color of light is being emitted by each one of those dots, but our brains perceive color differently. So maybe you've sat around thinking the age-old question, is my blue sky the same as your blue sky? Do we both see the same color blue? The answer is probably not. So the blue sky, the sky is radiating, is reflecting the same colors of light, and we can measure that. But our brains, each of us has a unique perception of color. So signals that arise from these 4 different types of photoreceptors are processed highly even before they reach the optic nerve. A lot of information filtering occurs where important information is highlighted, so to speak, and unimportant information is filtered out, so to speak. Our eyes and our brains are very sensitive to change, and they tune out the static or the unchanging conditions in our external environment. So if you look at the flow of information from photoreceptor to bipolar layer to ganglion cell layer to optic nerve, that's several layers of cells by which this information from the photoreceptors gets filtered, gets processed. So what we're seeing is not all of the information available. The information, rather, is filtered before it even makes its way into our brain. And then once that information makes its way into our central nervous system at the optic chiasm, that visual information travels along many different visual pathways. And the pathway that is responsible for your conscious awareness of what you see occurs as part of your occipital lobe. That's part of your cerebrum in the posterior aspect, the back of your head. Your cerebrum includes your primary visual cortex. But there are there are dozens of different brain regions dedicated to processing visual information. Notice that some of that visual information goes through your midbrain. For example, the superior colliculus is part of your midbrain. And I'll remind you that we did talk about visual reflexes, including the pupillary reflex, the accommodation reflex, and visual tracking. So your midbrain processes visual information and you have no conscious awareness over it. If you're curious, you should look up a phenomenon known as blind sight. And people with blind sight are, for all practical purposes, they are blind. If you hold an object in front of their face, they won't be able to identify it. They won't be able to see the pen or the coffee cup. But if you have them walk down a hallway, they are able to successfully navigate around obstacles. So they walk down a hallway, they can walk around the potted plant, they can avoid chairs that are in the hallway without really understanding at a conscious level how they are able to navigate around obstacles. And so the current hypothesis is that a lot of that visual information is processed in their midbrain, even though their primary visual cortex is not working. Their midbrain and their reflexes are working. And without the involvement of the cerebral cortex, they don't have any conscious awareness over it. It's called blind sight. It's super interesting. These are other exhibits at the Exploratorium dedicated to light and perception. So that little boy, the yellow little boy, he is in what's known as the monochromatic room. And so that that room is very colorful, but only when it's illuminated in white light. And when you walk in, it's illuminated in a pure yellow light. And because the only wavelength of light available is yellow, that deprives the objects the opportunity to reflect the other colors of light. And so those other colors of light are just not present in order to strike our photoreceptors, our opsins. And so we have no color perception in that room. But notice that he's holding a flashlight. There are little flashlights hanging from the wall. And in the white light produced by that flashlight, we can see that there's some blue in his shirt, for example. And there's a gumball machine full of jelly beans there. And only using the flashlight can you see the different colors of the jelly beans. All right, moving over to the top right, I think this is one of my favorite exhibits at the Exploratorium. I know I've probably said that 3 times already. I don't know the name of this exhibit, but as an introvert, I really like it. You sit down in the chair and you pull this helmet over your head. So you're suddenly in your own world. In this busy, bustling museum, you can kind of be by yourself for a little while. And so you have this globe of a helmet around your head, and on one arm of the chair is a dial. And as you scroll through that dial, as you turn that dial, the color of light emitted by the helmet, the color of light that is enveloping your whole head changes. You can scroll from pink to orange to blue, and you can check in with your body and see how each color makes you feel. And then down below, this is a pretty common neurological test, actually, where your goal is to name the color, not read the word. So sort of in the middle there, I'll read the words. It says yellow, lavender, purple. And instead of saying those words, your goal would be to say the corresponding color, red, black, green. It's quite difficult. And then on the lower left, we can see color broken up into its fundamental components. That's magenta and yellow and cyan, and they're making really cool shadows. After images are fantastic demonstrations of receptor fatigue. So in class, hopefully it's a dark classroom, and this image is projected on a large screen in front of the room. And I ask students to just stare at this green square with the red word after image, and students will stare at that for perhaps 30 seconds. And after 30 seconds, I change the slide, and it's a white slide, so it's like a white background. And most students in the class report that they do see an after image. And what an after image is, it's almost like the negative of the image that you've been staring at. For those of you who are old enough or experienced enough to know film, in the old-timey cameras where there was film involved, you'd take a film and then you take a photo and then you'd have the film developed. And there would be such a thing called a negative for each photo. In the after image, what it's caused by is the fact that photoreceptors become fatigued. So as you stare at this color green and this color red for 30 seconds, those various photoreceptors that respond to these wavelengths of light, they're firing, they're firing, they're firing, changing shape, ions are flowing. Meanwhile, the other opsins, the other photoreceptors in your eye that don't respond to these wavelengths of light, they're quiet, they're just sitting there bored, they're not doing anything. And so after you move over to the white background and see the after image, your eye is then at that point taking in white light, which includes all the visible spectrum. And so the photoreceptors that were tired or fatigued by staring at this green image with the red writing, those photoreceptors fail to fire. And so what you see when you look at this all white screen is the after image. So it is the absence of those photoreceptors firing. And here's that white background. Sensory adaptation. Sensory adaptation is another way in which your nervous system is quite tuned to respond to changes in our environment and to not respond or pay much attention to aspects of our environment that are fixed or constant or static. So you have experienced sensory adaptation today, every day, but here are some examples. So let's say you walk into a classroom and there's a buzzing light fixture and you're like, gosh, that's annoying, but you sit down and you get ready for class and 10 minutes later, you don't even notice the buzzing light fixture anymore. Or you walk into a classroom and somebody left their lunch there and it's tuna fish and you hate tuna fish and you don't like that smell, but you have class and so you need to stay in the room. 10 minutes later or so, you don't even notice the scent of tuna fish anymore. This is due to sensory adaptation. Another example of sensory adaptation comes when we're eating food. If you're a food lover, maybe you have wondered, well, what is the most delicious bite? You're eating a dish that maybe you prepared or maybe somebody prepared it for you. You're really looking forward to it and you have your first bite. Is that the most delicious bite, or is it going to be the second bite or the third bite? Or maybe it's the 10th bite. What is the most delicious bite of that meal? Well, sensory adaptation influences our perception of taste. And here the example is wine. And you are most sensitive to taste earlier in your meal or earlier in your glass of wine, for example, or your beer or your glass of apple juice, where we can see on the vertical axis is perceived intensity of flavor, and then on the horizontal axis is increasing number of tastes. So your first taste is the most intense, and then that intensity declines with every subsequent taste because your taste buds undergo adaptation. So during a constant stimulus, like eating the same bowl of macaroni and cheese, with every bite, it's the same macaroni and cheese. That's a constant stimulus. The graded potentials that are generated by those taste receptors will gradually decrease in amplitude. So remember, graded potentials are not all or nothing. Graded potentials can be depolarizing, they can be hyperpolarizing, they can be big, they can be small. So if taking that bite of macaroni and cheese generates in particular taste receptors, generates graded potentials, over time it will generate smaller and smaller depolarizing graded potentials, and that will lead to fewer and fewer action potentials. And so as our taste receptors, as we're using as the example here, as our taste receptors are exposed to the same stimuli over and over and over again, ion channels gradually become inactivated. So the same stimulus will no longer open the same number of ion channels. So that will lead to a lower amplitude of graded potential. Here's another example of our sensory receptors undergoing adaptation. So have you ever gotten into a body of water? You've gone swimming in a pool or the ocean or a river, and somebody says, "Hey, how's the water?" And you would say, "It's nice once you get used to it." But what you're really saying is it's nice once your thermoreceptors undergo sensory adaptation. Our second special sense that we'll cover as part of the nervous system is gustation. Gustation is your sense of taste. And you have a sense of taste because you have chemically sensitive cells that occur in these structures known as taste buds that are part of your tongue and your pharynx, part of your throat. And these chemically sensitive cells are able to detect and generate graded potentials that lead to action potentials in response to molecules or ions that are dissolved in your saliva. There are 5 primary tastes. Perhaps you're familiar with them. What are the 5 primary tastes? Maybe you learned about them in 3rd grade. Maybe you learned about them last week. The 5 primary tastes are sweet, salty, bitter, sour, and umami. And umami is the most recent discovery. Umami means savory or good flavor in Japanese, and it was discovered, this particular receptor was discovered by Japanese researchers. And so salty taste receptors respond to dissolved sodium and chloride. Sweet responds to dissolved monosaccharides like glucose and fructose. Bitter responds to dissolved plant alkaloids. This is why so many drugs have a bitter flavor. And sour receptors respond to low pH, like lemon juice is sour. And then umami responds to glutamate, dissolved glutamate. Glutamate is an amino acid, and amino acids are abundant in protein-rich substances like meat. Although it's my understanding that meat isn't the only thing that can have an umami quality. Mushrooms are umami, seaweed is often described as umami, tomatoes are often described as having umami, and they are high in glutamate. We have just 5 types of gustatory cells: sweet, salty, sour, bitter, and umami. But we can detect thousands of flavors. What's the difference between a strawberry and a banana? What's the difference between, I don't know, a margarita and chocolate? So just like with vision, a lot of processing occurs as those sensory receptors detect these different chemical substances dissolved in saliva. There's a lot of post-detection processing that occurs before that information is even sent to your brain. And then, of course, perception occurs in your cerebrum. And that location is in the gustatory cortex, which is part of your cerebrum. It's number 5 there in the image. So I enjoy wine. I like to go wine tasting, but I have learned to only drink what I like, even if it's a cheap bottle of wine that somebody else might be embarrassed by. I have tasted very expensive bottles of wine that I frankly don't care for. And of course, I don't have that kind of money and I would never spend it. Every once in a while, I'll spend $20 on a glass of wine. That's really lavish for me. But there is abundant evidence to support the idea that wine tasting is rubbish. They do all of these studies where they do blind tastings, and the research subjects are people who are very knowledgeable in wine. So they are sommeliers, they are wine critics, they are judges at wine competitions. And when presented in blind tastings, they'll rank the wines. And then the very next day, the exact same wines are presented to the exact same judges, and they'll give totally different ratings of the wine. And so study after study has shown that by and large, wine tasting is bogus. There's nothing particularly special about a $70 bottle of wine versus a $5 bottle of wine. And so just drink what you can afford and what you like. This brings us to the end of our coverage of the nervous system. This is the end of segment 5 of 5 recorded lecture segments about the nervous system. And today, in this recording, we covered topics 6 and 7. We talked about neuroplasticity and just 2 of our special senses, vision and gustation. I will see you next time.